Hi class. Oh, Alex, you're back again.
Yes, ma'am. I really need some knowledge on action potential as well.
Sure, but action potential is a very lengthy topic, so you might have to sit more in this physiology class.
Anything for the knowledge, ma'am.
Perfect. So, students, the topic for today is action potential. Remember in our last lecture, we talked about resting membrane potential. Can anyone tell me what was that?
Resting membrane potential is the state of the cells when they are in a non-excited state.
Yes, and the action potential is the state of cells when they are in an excited state. Can anyone also tell me that what was the charge on the cells when there was resting membrane potential of the cell?
There was a negative charge on the cells. That means intracellular space was negatively charged and the extracellular space was positively charged.
That is right. But in the case of the action potential, there will be more positive charge on the intracellular space and more negative charge on the extracellular space. And, does anyone remember that which ion channel was mostly involved in resting membrane potential?
Both sodium and potassium were involved in the resting membrane potential, but the major roles was of the potassium ions and involvement of sodium was less.
Absolutely correct. And in case of action potential, there will be involvement of both sodium and potassium ions, but this time, sodium will be taking the lead. Lastly, can anyone tell me about the channels involved that were mainly causing normal resting membrane potential?
Yes, there were sodium-potassium pump and potassium leak channels for the establishment of resting membrane potential.
You are so correct, Jenny. In case of action potential, there will be activity of sodium and potassium channels majorly, but more of sodium channels.
That was an easy comparison of both the membrane potentials.
Yes. Just learn the one membrane potential and the other one works the exact opposite of that.
So how does action potential start?
Let me tell you that. When you looked at the typical cell membrane, there were two channels involved in the resting membrane potential: sodium-potassium pump and potassium leak channels. Now there are other two channels located on the cell membrane that are known as voltage-gated sodium channels and voltage-gated potassium channels. Both of them are responsible for the action potential. What was the voltage on the resting membrane potential, by the way?
It was minus 70 millivolts.
Yes, so whenever the voltage moves from minus 70 to the value of 0, action potential will start through voltage-gated sodium channels.
But there are also ligand-gated channels in the cell membrane. Why are we not talking about them?
This is because in action potential, there is the change of voltage and not the attachment of a ligand. So only voltage-gated channels will be working when we talk about action potential.
That makes sense. Please tell us about both voltage-gated channels.
The first one is the voltage-gated sodium channel. This is what a voltage-gated sodium channel looks like. It has two gates, one we name as activation gate, which is present at the outside of the cell membrane. The other one is the inactivation gate, which is present on the inside of the cell. Consider both of these gates as the lid of the jar which is attached to that jar. When you open the lid of the jar, what happens? You can easily get the inside jam or chocolate out, and also if the jar is empty, you can put any jam or chocolate inside the jar. The same is the case with these gates of sodium channels. Only that this jar has lids on both sides, from the top and the bottom. Now, one more special thing about these channels is that one lid will be open at one time. It will never happen that both lids will be closed. There might be a case where both lids of gates may open, but the total closing of gates will never occur.
I have a question. You said that they are sodium channels. Then how does that channel know that I will allow only sodium to pass through?
Very good question, Jenny. This is due to the selectivity filter on each of the channels. Sodium gates will have a selectivity filter that will only allow sodium to pass through. And same is the case with potassium. I would love to tell you about the working of selectivity filter, but that is the topic for another day. Coming back to our topic, which was the voltage-gated sodium channels and their gates. Now at the resting membrane potential, what do you think will be the condition of sodium channels?
I think that the activation gate will be closed due to no influx of sodium inside the cell.
Yes, that is right. And then what will happen when the resting membrane potential starts to move towards the value of 0 from minus 70 millivolts? The activation gates will be opened, and the sodium will start coming inside the cell. This will happen continuously until the negativity of the cell will be converted into positive. As there is sodium coming continuously inside the cell, there will be the creation of a positive charge on the inside of the cell.
And what would be the potential difference at which there is a maximum influx of sodium?
The average value is positive 35. But it may vary depending on the type of cells that have action potential. For some of the cells, which are small nerve fibers, the influx of sodium is stopped even when they reach the value of 0 and most positive. Anyways, now we have achieved a positive charge on the inside of the cell. Can anyone tell me what will happen next?
The activation gates will be closed due to enough influx of sodium?
No, the inactivation gate will be closed like the same way activation gates were open. This is because activation gates take time to close and we need to terminate the influx of sodium quickly. So, the inactivation gates will close faster.
So, will the activation gate be closed as well?
Yes, the activation gates will completely close when the resting membrane potential will be achieved. Now remember one more channel that was involved in this action potential. That was a voltage-gated potassium channel. If we look at their general structure, there is only one gate, unlike sodium channel, which is known as the activation gate. This activation gate is located inside the cell. What happens is that when there is too many positive ions inside the cell wants to get on the state of rest once again and wants to get this positive charge out of it. Now for doing this, it will open the activation gates of potassium. After the gates are opened, the potassium will start getting out of the cell to manage the positivity and move towards the state of rest.
All right. So, sodium channel will cause the positivity inside the cell, and potassium channels will balance that positivity by getting out of the cell.
You are right. These are the major role of these voltage-gated channels. Now there are different states of the action potential which we will be discussing next, and we will also relate those states with the activity of these voltage-gated channels as well.
Ditch the hefty textbooks and embrace the convenience of learning on the go. Our interactive video content keeps you engaged, making learning medicine enjoyable